Good morning, students. Today we will study Monera. According to the 5 kingdom classification of RH Whitakers. Well, Monera is exclusively a prokaryotic kingdom. Prokaryotic kingdom means the cells or the life form, the unicellular life form, have nucleus of primitive form. Primitive form means nucleus is not well organized. Genetic material is directly embedded in the cytoplasm. The term moneris was first used by Ernst Haeckel for a phylum under the kingdom Protista. Monera is exclusively prokaryotic kingdom. It is divided into subphylum on the basis of phylogeny, on the basis of their chemistry, structural organization, on the basis of their cellular processes like transcription, replication. The Monera is divided into two major groups. The one group is called Archaea. Monera can be divided into two major groups. One is called Archaea and the other group is called Eubacteria. Eubacteria. Why is it divided? Carl Lewis studied that there is a group of monerans which is different from another group in their morphology and physiology and also on genetic level. The main genetic basis of this division is 16S rRNA gene. 16S rRNA gene is different in these groups, and this is the solo basis of division of monerans. Other differences between Archaea and Eukarya is the cell wall. Here the cell wall is made up of pseudopeptidoglycan. And here the cell wall is made up of peptidoglycan. Another difference is in their cell membrane. The lipids are ether lipid in Archaea. Ether lipid are present in cell membrane, whereas the lipid present in cell membrane is ester lipid. Another difference is it does not form spores. no spore formation reported. A spore formation occurs. So, inside the kingdom Monera, there are two phylum. One is Archaea and the other is Eubacteria. They are different from one another genetically, solo based upon the organization of 16 srRNA genes. Other differences which are visible is the constitution of their cell wall, constitution of their cell membrane, some lifestyle habits, and also the processes like transcription, replication, machineries are different from one another. They are differently susceptible and have different reaction with different type of enzymes and antibiotics. So, kingdom Monera has two phylum, Archaea and Eubacteria. Otherwise, we can also, these are not phylum, these are organized as sub-kingdom. Archaea is a sub-kingdom and Eubacteria is a sub-kingdom under the classification kingdom, 5 kingdom classification, in which Monera is a kingdom and these two are sub-kingdom. Under this sub-kingdom there are several phylums. Approximately there are 24 phylums here in Archaea, and there are more than 50 phylums are under the sub-kingdom Eubacteria. But very less have cultured representative. Among 24 phylum, only 8 phylums are represented by a species here. Okay. Now we see the types of Archaea. Archaea, as we have said that there are more than 24 phylum, but only 8 has been reported with a species. They are autotrophs as well as heterotrophs. Most of the group of Archaea, which is categorized on the basis of their nature and the environment they live in. So the major groups of study today in Archaea are methanogens, thermophiles, halophiles, These are not the phylum, these are the groups. They may present from different phylum. These constitute extremophiles. Extremophiles. That means they are the group which survive in hostile conditions. And such groups are named as extremophiles. Among these extremophiles we have methanogen, thermophiles, halophiles. Who are methanogens? Those who produces methane gas are called methanogens. Example: methanococcus, methanobacter. Example: methanococcus, methanobacter. Okay. Now, these methanogens has been reported from the intestine of ruminants like cow, buffalo, and the cow dung also contains methanogen bacteria. That's why it is used as inoculum in biogas plants or septic tanks. Now, thermophiles. Those are key bacteria which survives in hot winds, springs, where the temperature is 80 degree, 90 degree centigrade. In such hostile condition, the archaic bacteria are called thermophiles. The bacteria, archaic bacteria, which can survive in extreme temperature. Example: Thermobacter, Thermococcus. Now, halophiles. Those who survives in extreme saline condition. Now we come over the nutrition in archaic bacteria. Nutrition. It has been reported in archaea that they utilize inorganic as well as organic source for production of food. But not a single species has been reported which can do both. Okay, so they are phototrophs, they are heterotrophs, they are in heterotroph. There is no any form which has been reported as pathogenic to human, but they have been reported as symbiotic life forms like methanogens. Nanogenesis. It's a symbiotic life form. Reproduction in archaea occurs by asexual means, that is binary fusion. A spore formation has not been reported. So this is all thing about archaea. Okay? In next class we will study about eubacteria in detail. So I just conclude this class by the kingdom Monera is one of the 5 kingdom classification by R.H. Whittaker. Monera is specially prokaryotic kingdom in which life forms are unicellular and they are prokaryotic cell. That means nucleus is not well organized. Nuclear membrane is not present. Nuclear membrane is not formed over the genetic material. Genetic material is directly embedded in the cytoplasm. On the basis of 16S rRNA, Monerans have 2 groups. On the basis of 16S rRNA, Monerans have 2 groups. These groups are named as archaea and eubacteria. Archaea, whenever we say archaea, then it is archaebacteria and eubacteria. These are the 2 groups. These 2 groups have morphological, physiological differences from one another. Cellular differences of cellular processes also lie there. What are the morphological differences? Morphological differences are their cell membranes, their cell walls are different. One is pseudopeptidoglycan, other is peptidoglycan. The cell membrane of archaea is made up of ether lipid, whereas the cell membrane of eubacteria is made up of ester lipid. Again, the cell membrane of archaea contains sterols, which is not present in eubacterial cell, but present in eukaryotic cell. Then the difference lies in cellular processes like transcription, like replication, machineries, and the processes are different in this 2 groups. So, in archaea, there are 24 phylums. On the basis of 16S rRNA genes, this phylums has been created. But only 8 phylums is represented by a species. Okay? Now we come over the nutrition. They are autotrophic as well as heterotrophic. In autotrophic, there may be phototrophs as well as chemotrophs. Now they reproduce by binary fusion. So this is all about archaebacteria. In next class we will study about eubacteria in detail. Thank you.